For chapter 3. This is going to cover the chemistry of organic molecules. Overall, we're going to look at carbohydrates, lipids, proteins, and nucleic acids, but we're going to divide it up a little bit. So part 1 we're going to cover in this video, which is going to set the stage, talking about organic molecules in general, and then we're going to go into carbohydrates. So yeah, organic molecules, carbohydrates, sections 3.1, 3.2 in your textbook, and then 3.3 through 3.5 we'll cover in the next presentation. Just to set the stage a bit, we often hear of trans fats, which is a type of fat that is actually like man-made. We are adding hydrogen to an unsaturated fat in this process called hydrogenation. So you've probably heard in the news about trans fats and hydrogenation, hydrogenated soybean oil, and things like that. These are all things that we do to chemically enhance our food. They have the benefits of increasing shelf life and maybe improving or altering the flavor or texture of the food, but we're starting to find out now that perhaps they, even though they are just a type of fat, not all fats are created equal, and they might be having more adverse effects on people's diets. So for that reason, you see that things are usually labeled as having trans fats in them. In fact, most things now are even being labeled as trans fat free because people recognize that there's a good likelihood of negative health consequences to trans fats. That's just one of the ways in which this sort of thing would affect your daily life. Just looking at those types of fats that you eat. Obviously, all these molecules can relate very easily to food and what we put in our bodies, and then just keeping your body going. And so they can relate to you like on a a social level, just thinking of what you're eating, but also just thinking about how your body is working. So organic molecule is any molecule that contains carbon and hydrogen atoms, right? That's the key. So for example, like carbon dioxide, CO2, contains carbon, but it doesn't contain hydrogen, so therefore it's not organic. Generally, these are things that are going to have come from living materials at some point in time. One obvious thing to keep in mind is that organic can have different definitions, right? We think of organic from the grocery store. Organic foods are things made without synthetic pesticides. Their roots are the same, meaning like natural or living, but it's a little bit different. The four main organic molecules that exist in living organisms, things we're going to be looking at: carbohydrates, often just called carbs, lipids, proteins, and nucleic acids. And they do a wide range of things in our bodies. One thing when you're thinking about organic molecules, a lot of times we talk about all these functional groups. We'll talk a little bit more about those later, but this is a handy chart just to help you see what all the different functional molecules are. And we'll talk about hydroxyl groups, that's having an OH, or carbonyl groups is a carbon double bonded to an O with an H. Carboxyl group is just a double bonded O. Also, like those would also be alcohols. Amino groups, NH2, so like amino acids later on. Phosphates we'll definitely be talking about. So that's a phosphate group, PO4. So these are all some different groups that you might see and encounter as we're looking at pictures of these different molecules. So what's so special about carbon? All this stuff is made up of carbon. We talk about life on Earth being carbon-based. It really is. It's part of everything living. And the cool thing about it is because it's a small atom, relatively few electrons, and we look at the structure of our carbon, we see that the valence electrons has 4 valence electrons, which means it's kind of in that position where it doesn't necessarily want to give up electrons, it does want to get some more, so it tends to form covalent bonds really easily, and it tends to form up to 4 covalent bonds really easily. So it can bond very easily with carbon, nitrogen, hydrogen, oxygen, phosphorus, sulfur, all those elements that we talked about in our chapter 2 notes, those elements of life. It's a very stable bond, and you can form tons and tons of different forms, right? So the simplest, I guess you could say, might just be something like this. So you've got a carbon bonded to some more carbons. This one's bonded to hydrogen. All right, just a simple form. You can have double bonds, right? So you're actually sharing 2 pairs of electrons here. You You could have triple bonds where you're sharing 3 pairs. You could do these square shapes, and you can also do these ring shapes where you're alternating double bonds and single bonds, which are very, very common. These are called aromatic rings. We see those in lots of different organic molecules as well. So a wide variety of shapes, which then allows a wide variety of molecules to do a wide variety of functions. And so when we talk about these molecules, we talk about the carbon chain, or the main backbone of the molecule. So sometimes it's a long literal chain, sometimes it's that circle. But on those, that's sort of like it's the backbone, like that's the common part. But then there are functional groups, and that's what I was talking about before. So you could have a long line of carbons and hydrogens, and that's great. But the functional groups are the little bits that are on there that can, just like it sounds, alter the function. So if it's an amine group, maybe you're making an amino acid, or a hydroxyl group. You could be making a base, or a hydroxy group, and you're making an alcohol, right? So you can get lots of different things there. So those functional groups determine what it's going to react with. It's going to determine if it's polar, right? And we talked in the last set of notes about polarity and how that polar molecules can interact with different things that nonpolars can't. It's stronger bonds, things like that. An isomer is a type of molecule. They have identical formulas, but just the arrangement of those atoms are different. So if we look at these two, for example, both have 3 carbons, both have 1, 2, 3, 4, 5, 6 hydrogens and 3 oxygens. We're just changing the location. So in this case, your double bonded O is in the middle. And so these guys can have totally different functions, even though their chemical formula is exactly the same. The way they're put together gives them different jobs. So those are isomers. Iso means the same. And mer comes from polymer, so the same polymer. So these molecules that we've been talking about, the carbs, lipids, proteins, nucleic acids, are often referred to as biomolecules because they're important for life. They're made up of repeating units called a monomer. So this is like the subunit. So like it's almost like the bricks of a brick wall. And so, for example, in this polymer we're working on here, this is your monomer, this chain, and then we link lots of those together. 1, 2, 3, 4 monomers linked together to form a polymer, many parts, and so you can string all those together. So all these biomolecules have a most simple functional unit, most simple part. And so, for example, amino acids is the monomer, and those join together to form a protein. That's the polymer, right? Lipids, technically speaking, are not polymers because they have 2 different types of subunits. So it's like 2 different monomers. So technically speaking, they're not polymers, but they are similar in a lot of aspects, and they're like long chains of many repeating units. And so if we look at these are our 4 molecules here: carbs, lipids, proteins, nucleic acids, and these are their monomers, right? So a monosaccharide is the monomer of a carbohydrate, and the polymer form is called a polysaccharide. Lipids are made up of glycerol and a fatty acid, and their polymers are called fats. Amino acids bond together to form polypeptides, and then DNA or RNA are made up of the subunits, the monomers called nucleotides. So it's like the A, T, C, and G. Well, it's that, the adenine, cytosine, guanine, and bonded to other parts too. So we're going to talk a lot about synthesis and degradation, specifically dehydration synthesis, or dehydration synthesis and degradation. And so this is when dehydration reaction is when you're joining some things together and water is produced during the reaction. Okay, so we can connect monomers together to do this. And so, for example, for forming starch by connecting a whole bunch of glucoses together. And so if we look at this diagram here, we have 2 different glucoses, and this one has an H just hanging out here, this one has an OH hanging out here, and we put those together near each other. Hey, H2O. Those guys can disappear, they can break off, leaving this O to bond to this spot and create a linkage, and now we have these 2 guys joined together, and now we no longer have glucose, we have something called maltose. It's no longer a monomer, now it's a dimer, in this case a disaccharide because it's also a sugar. Another way of just showing this like a schematic view, you can imagine you have your monomer, right? Lots of monomers bound together, and your OH there on the end, and we want to add it to something else. Okay, we will find something else that has an H hanging out, and we can put those two together to form water. And once those guys form the water, that basically leaves little sticky ends on the ends of those monomers for them to break out. So dehydration, because you're removing water from those reactants. The opposite of that is hydrolysis. So you're using water to break a bond. And so it's going to break down your polymers into monomers or into smaller units. So an example is digesting starch into glucose, right? So any carbohydrates that you eat, so think of starch like in a potato chip, your body is going to break that down into glucose, into sugar that it can use for energy or that it can rearrange to make something else later. And so the same idea in reverse, here's your molecules together, and you have that water, and that you can add that water in, where one of the H's is going to go with the O that's hanging out there. The other OH is going to go to that other sticky end, and now you've got two monomers, and you've used up that water to break those guys apart. So simplified version, again, I didn't show all the little steps, but same idea. You're going to break these guys apart by adding water, and that water is going to split. The water is going to be is split. That's why it's hydrolysis. Lys means to break, hydro, water. So you're breaking that water apart into H and OH. So there's both the processes together in one diagram. So a lot of times this is done, it doesn't just happen by magic. It happens with molecules called enzymes. An enzyme is a molecule that can speed up a reaction that would already happen. So if left to its own devices, this would happen. It would just take a really, really long time. In most cases, too long to be biologically relevant. It needs to happen faster. So an enzyme does that job. Some important things to know about enzymes is they're not used in the reaction. They help the reaction happen, but they stay intact, right? So they're not consumed and they're not changed. And we also refer to enzymes as catalysts, right? This is something that helps that it starts the reaction. A catalyst is something that provides a jump start, let's say. Right? So maybe you've heard of the term catalyst in other non-scientific contexts, but in this case, it's going to start that reaction. And just an example, these three pictures that seem very, very, very different actually have a lot in common in that they are all utilize carbohydrates in lots of different ways. So a plant has a cell wall made of cellulose, which is a large chain of carbohydrates. This guy here, this crab, this shell contains chitin, and so it feels real hard, and that's actually another carbohydrate in a different form. And even something like this bacterium has, in its cell wall, has something called peptidoglycan, just a different form of carbohydrate. So we can take those carbohydrates and we can put them in lots and lots of different orders and make lots of, like a very diverse amount of chemicals for a diverse amount of functions, from a tiny bacterium to a giant saguaro cactus. So carbohydrates specifically. Mostly we think of these as an energy source. Carbohydrates can often be a quick energy source. You think like sugar rush. And what they can also provide building material. So we just saw that in our examples of our chitin, and we saw that with the cellulose. That's providing a structural role. Or energy source in, like I said, glucose or starch, right? Potato is really just like the potato that you eat is just the energy storage part of that potato plant. These guys contain carbon, hydrogen, oxygen atoms in a 1 to 2 to 1 ratio. So just as many carbons as oxygens and twice as many hydrogens. We talk about monosaccharides, disaccharides, and polysaccharides. We don't usually talk about like a trisaccharide, three of them. In that case, it would just become a polysaccharide. Mono being 1, di being 2. So monosaccharide, just a single sugar molecule, 1, called a simple sugar, usually a backbone of 3 to 7 carbon atoms. Common examples of these are glucose, which is our blood sugar. Fructose is the sugar that's in fruit, so it's what's making your apple sweet. And galactose is another example. These guys come in a couple different forms. You see these up here, a couple different shapes. You've got your 6 carbon sugars. These guys form a hexagon, so they're hexoses. This is a pentagon, 5 sides. That's a pentose. Other common examples of pentose are ribose and deoxyribose. Maybe you remember those whenever we've talked about nucleic acids in the past. Some different ways of picturing these molecules, right? These are all C6H12O6. These are all glucose. You can number and have all your different carbons shown here, showing your OHs, right? These guys are all bonded together. This shows everything. Shorthand would be you're not going to list all the numbers, right? So that's not really that much of a shortcut. You could also do this and shows you, basically this is just showing you that backbone, that you have this oxygen here. All these other vertices, all these other corners are carbons, right? If they don't label it, we assume that it's a carbon. And then this shows that you got something else bonded there. And then like if you really didn't care necessarily about all those extras, you might just show it just like this. This is probably the most common format of way something is shown. Or maybe like a combination of both. Maybe it might have this, but only show like this group here because everything else is sort of boring, like, yeah, OH repeating, like no big deal. Disaccharide, just 2 monosaccharides joined together by a dehydration synthesis. So some examples of those: sucrose, that's what's on your table sugar. That's a fructose bonded with a glucose. Maltose is 2 glucoses added together. Lactose is a galactose and a glucose put together. That's what you find in milk. And so lactose intolerant individuals don't have the enzyme that's going to break that lactose down, which is so they don't digest it very well, which is why they end up with gas. Just to show that reaction, I'm going to go through this one step by step again. So if you're looking at this glucose, your 2 glucoses together, we've got our OH and our H highlighted together, and we can form some waters with those by dehydration. So we'll pull out the water, bond those together, you got your water hanging out. So you got a monosaccharide plus a monosaccharide turning into a disaccharide with some water left over. Similarly, you could break those down. So here's our maltose, and we're going to break that apart. We could add some water to that. We're going to break up that water, and it's going to go back the other way. So we're going to the left now. And so you add that water in there, and that can go into help break that apart and split these guys. So just again showing that these guys are the same thing in reverse, right? If you're going this way and putting things together, it's a dehydration. If you're breaking things apart, it's a hydrolysis. Okay, same reactions, just forwards and backwards. A couple of more starch or other carbohydrate molecules. Polysaccharide, again, is many monosaccharides joined together. So far we looked at the disaccharides. Starch is a common one. I talked about that already in potatoes. That's energy storage in plants. Glycogen, similar to glucose, right, in word-wise, but glycogen is a much more complex molecule that provides energy storage for animals. In cellulose, we've talked about that a little bit already. That's found in the cell walls of plants. Most abundant organic molecule on Earth, and we're actually unable to digest this. This is why we talk about eating fiber to stay regular, because that fiber just moves through your digestive system and keeps things moving. Animals that do eat a lot of cellulose actually get help from bacteria inside their gut that are going to actually do the work of breaking that down. Chitin, we saw that already too, the cell walls of fungi as well as exoskeleton of a lot of animals. And peptidoglycan, also showed you that at the beginning of this little section, in the cell walls of bacteria. So building together different things. So lots of different polymers with lots of different jobs. They could be structural, like these guys, or energetic, like these guys. Well, I guess this is more structural. These are energetic. Just visualizing these a little bit, starch molecules. So here's your potato, right? This is what it might look like with a scanning electron microscopy. And so you actually have these long branched chains. Amylose is starch, and so really what you've got is this, like lots of these glucoses all joined together, and then they start to get this branch structure. As you sort of zoom out, you see that. Here's glycogen down here, often found in the liver. That's why we're showing it here. And you can see just a string of these same glucoses, right? But much, much more complex and crazy branching patterns. But same base units, right? Just rearranged and connected in different ways. Here's another one, cellulose. This one is more fibrous, and so you end up just with lots of long strands of these, and what's holding them together? Ooh, yep, hydrogen bonds. We got some hydrogen connected to that oxygen, so that's going to help keep those together in that weak attraction, but help bond all those little fibers into one bigger fiber, which then forms like a lattice that's going to form a plant cell wall. All right, and that is the end of that section of notes.